Hello and welcome to this session of study that we're going to study today about cell division. We know that reproduction is the basics of life and how life continues. And in order to understand, we should also get to know more about cell division and how it takes place, what are the various stages, and how division actually takes place in plants and animals. Cell division from a mother cell to daughter cell can give 2 daughter cells or 4 daughter cells, as discussed in the 10th class of biology, that it is divided into mitosis, where cell division gives rise to 2 daughter cells, and meiosis, where it gives 4 daughter cells. And we know that every cell is capable of divisions and how these events, which are called as cell cycle. The sequence of events including duplication of DNA, synthesis of other cell constituents, growth and division that a cell undergoes from the time of its formation up to its division into daughter cells is called a cell cycle. And the gap between first cycle or a second cycle, that gap is called the generation time. A cell cycle consists of the 2 following phases. Number 1 is interphase and second is mitotic phase. Interphase is a non-dividing phase, whereas mitotic phase is a dividing phase. And interphase is also often called as the preparatory phase, where a cell is actually preparing for the cell division, which is going to happen in the mitotic phase. Interphase, it is where, as you can see in the diagram, how it is divided into 3 sub-stages. First one is G1 phase, and secondly S phase, and third is G2 phase. G1 phase, which is often known as the first growth phase, that is why it is termed as G1 phase. In this phase, what happens is that the RNA and proteins are synthesized and the overall volume of cytoplasm increases, hence the cell grows in size. There is no change in the DNA contents of the cell during this phase. So now the cell moves to S phase, which is also known as synthesis phase. And in synthesis phase, synthesis of DNA takes place and the chromosomes get duplicated, or we can say it makes its own copy. And third comes the G2 phase, which is known as second growth phase of pre-mitotic phase. In this phase, it's a very short phase and synthesis of DNA stops, but synthesis of RNA and proteins necessary for cell division continues because it is preparing for the cell to divide. So now comes the mitotic phase. Mitotic phase, which is also known as the dividing phase of the cell, and during this phase the nucleus as well as the cytoplasm divides into 2 daughter cells, undergoing 4 important stages, which are Prophase, Metaphase, Anaphase and Telophase. And we know that the production of cell and their death in our body happens every day. So in order to regain those lost cells, we need to gain, and that is why our cells of the body needs to replenish the lost cells. Now let us talk about the types of cell division. Cell division is of 2 types. Number 1 is mitosis, number 2 is meiosis. Mitosis, it is a type of cell division during which a cell divides into 2 daughter cells each containing similar and same the number of chromosomes as present in the parent cell. So since the number of the chromosomes are the same as the daughter cells that are derived from the mother cell, both of them have the same number of chromosomes, and this is why it is also known as equational cell division. And as it only takes place in the non-reproductive cells of the body, like repair the tissue and the lost skin cells and throughout the body, so it is also known as somatic cell division. And somatic cells are those cells which does not take part in sexual reproduction. Okay, now let us see the diagrams and focus on the various stages how this mitosis, which is the equational cell division, also known as somatic cell division, takes place. Number one is prophase. In prophase, it begins with the shortening and thickening of chromosomes. Centrioles in the centrosome of animal cell divides into two, and then what happens that these centrioles develop a very fine eyelash-like thread called aster rays. The two aster start moving toward the opposite poles. By the end of the prophase, they reach at the opposite poles, and between the two asters develop a very fine thread-like structure called as spindle fibers. The chromosomes get attached with the spindle fibers from their centromeres, and the nuclear membrane and nucleus start disappearing in the middle of the prophase and totally disappear by the end. So here first the chromosomes, they shorten and they thick because the condensation of the chromosome takes place, and they come out of the nuclear membrane as the nuclear membrane disappears. And here the prophase ends. Next stage which comes is metaphase, and in metaphase the chromosomes begin to show peculiar movements, and they arrange themselves between the poles on the equator of the spindle, and they form a equatorial plate. So as the cell, in between the cell, they form a equatorial plate, and they arrange themselves, and they also get attached along with their centromeres to the spindle fibers. Next stage comes as anaphase. In anaphase, each centromere splits into two daughter centromeres, each retaining one of the two chromatids, which are known as sister chromatids. And they start moving towards the opposite poles, and the movement perhaps is brought by the contraction of the spindle fibers. So as they are attached with the centromere of the sister chromatids, and they split, and their main motive is to reach the opposite poles, and by doing so the contraction of the spindle fibers force them to move towards the opposite poles. As in the earlier stage of prophase, the centrioles have already reached the opposite poles, and by the end of the anaphase, out of each pair of sister chromatids, both of them reach to the opposite poles, one at the other end, the other at the other end. Next stage, which is the last stage of mitosis, comes and is known as telophase. In telophase, each sister chromatid, which is the daughter chromosome, which reaches at the opposite pole, starts uncoiling, thinning and elongating. because they form a network-like structure called chromatin reticulum. And in this stage, when the chromatic reticulum is formed, then the nuclear membrane reappears and they give rise to two daughter nuclei. And in the two daughter nuclei, nucleoli reappear in the nucleus, spindle fibers, ester rays disappear, and then they are surrounded by centromeres, again producing centrosome near each daughter nucleus. Up till this end, the mitosis is finished. But a very important phase, a stage which is coming, is known as karyokinesis. These four stages, prophase, metaphase, anaphase and telophase, are known as karyokinesis. And after this comes cytokinesis. Karyokinesis is known as the division of nucleus, and cytokinesis is known as the division of cytoplasm. So now in this cell, a furrow, a cleavage will appear, because as the two daughter nuclei, those small cells are ready to detach themselves from the mother cell, and so the furrow appears, a cleavage which will split the cytoplasm into two, and which will be called as cytokinesis. And when this happens, mitosis is finished. The cell division, this cell division will give rise to two daughter cells, and both of them will have the same number of chromosomes. As they are not taking part in the sexual reproduction, they are termed as somatic cells. Now let us talk about the significance of mitosis. It helps in the maintenance of chromosome number, which is a genetic stability. It maintains the cell of the size, as during in the beginning of the cell division it increases the cell size, so that when the two daughter cells are formed, they are of the equal sizes. And third point is growth. So when we are moving from a cellular level to multicellular level, when an organism grows, like for example a mango tree, from a seed it develops into a huge tree. So mitosis takes place every day to increase the size of that particular organism. Number fourth is replacement. So whenever your cell is damaged, you get hurt, or due to any kind of wear and tear in your body, mitosis helps in replacement of those dead cells, repair, regeneration and asexual reproduction. In repair, whenever a cell is lost, or whenever there is a wound on the surface of the organism, on a plant or in our own body, then mitosis helps to repair. In asexual reproduction, a part of a plant can be taken and the cells will divide continuously, and later in the stage it will form a new plant. Regeneration. Whenever you break a twig of a plant, then what happens is that part of the plant is regenerated by mitosis. And in embryonic development, the most important is embryonic development. So after fertilization takes place, embryo is there, it develops, so continuous cell division takes place in order for the embryo to develop into a fully grown infant which is ready, and then throughout the life cycle of that organism that it lives, and cell division, repair, significance of mitosis takes place. I hope that you have learned a lot from this chapter. The notes will be given in the description and you can take down the notes. And thank you. This is your teacher, Gagan Singh.